Hello, my name is Chester Kelly, and this is my video submission of my disease project that I did for my microbiology lab class with Dr. Swinson. I did my disease project over HIV, which is the human immunodeficiency virus. It's pretty well known worldwide. It's sort of an epidemic right now, has been since about the 1980s in the United States. And yeah, let's just get started. So, first slide. The HIV, or human immunodeficiency virus, is a lentivirus belonging to the family Retroviridae. The structure of the virus consists of two single-stranded RNA molecules, and they're identical, and they're enclosed within the viral core. The viral envelope contains certain proteins and enzymes that aid the virus in its invasion of CD4 cells of the body. So this is basically the structure of HIV. It has the viral core with the two pieces, or the two identical strands of RNA, and then on the outside it has different proteins around the viral envelope that help it fuse to the cell membrane and allow it to enter the cell. And then, also inside of the virus there is enzymes that it also uses to aid in this process as well. Second slide contains the life cycle of HIV. So the perpetual life cycle of HIV basically consists of HIV attacking the host CD4 cells of the immune system. CD4 cells are white blood cells pretty much. You can call them lymphocytes as well if you prefer that word. And these cells aid the body, your body, anybody's body, in fighting off infections. So whenever you get sick with the flu, whenever you get sick with pretty much anything, your body sort of launches an attack on the invader, and this is done with your CD4 cells. So pretty much HIV attacks the immune system, and this is what makes it so fatal if it, progresses to that point. So the first step of this life cycle is called binding. Binding is basically the attachment of HIV to receptors on the CD4 cells. This is just the attachment whenever it, first attaches to it. And then the second step is fusion. Using the proteins on the surface of the viral envelope, just like we talked about before, it uses these to fuse to the CD4 cell membrane, and this allows it to enter the cell. The proteins sort of act as keys that sort of unlock the cell membrane and allow the virus to enter the cell. The third step is called reverse transcription of HIV RNA into DNA. The virus uses the enzymes reverse transcriptase in order to enter the nucleus of the host cell to bind with the cell's DNA. And this is sort of what makes HIV so dangerous. Most viruses, they just kind of copy their RNA, but HIV, it reverses it. So it transfers it from RNA into DNA. And this is sort of what makes it so hard to treat, because it's very mutatable. It can change, and it's always changing, and it's just very hard to treat because of that. The fourth step is integration of the viral DNA with cellular DNA via the enzyme integrase. So integrase is one of three enzymes that HIV uses to replicate. The fifth step is the replication of HIV proteins using the machinery of the CD4 cell. So this is pretty much the part where, the virus sort of, invades the cell and uses the cell's organelles to kind of create its own copies of itself. And these are how these HIV proteins are created. And then that's where the sixth step of assembly comes in. And this is basically the assembly of new HIV proteins with new HIV RNA into immature HIV, which is non-infectious. And then the seventh step consists of the budding of immature HIV out of the CD4 cell. So after budding, these proteins, these HIV proteins, they're kind of broken down, and then they become mature, and that's whenever it's like a fully replicated form of HIV, and this is whenever it becomes mature or infectious. So the third slide is how is HIV acquired and where is it found? HIV is acquired from coming into direct contact with body fluids from an HIV-infected individual. These fluids can consist of blood, semen, rectal and vaginal fluids, and breast milk. HIV is also known as a sexually transmitted disease that can be spread through vaginal and anal sex. Anal sex is the biggest risk factor for it being spread sexually because of the lining of the inside of your body. It's very thin, and it allows for cuts to be made easily, and it allows these viral proteins or viral particles to enter the body on a very much easier level. Same with vaginal sex. It's not really transferred through oral sex or anything like that. Theoretically, it could be, but it's extremely rare. HIV is also commonly found in individuals who abuse intravenous drugs, which means like sharing used needles with each other that haven't been cleaned, or, you know, reusing them again after someone else has used them. It can also be found in gay or bisexual men who have sex with men and transgender women who have sex with men, because usually the way that, you know, gay and bisexual men have sex is the same thing we talked about before, you know, sort of anal sex, and that's the biggest risk factor for this type of infection. HIV can be found in anybody, regardless of sexual orientation, race, culture, or background. It's definitely not just a gay disease, which is a lot of stigma surrounding the virus. It can be, you know, transmitted and infected with anybody, you know. But people from the Hispanic, Latino, and Black demographics are also more affected by HIV than other demographics. And pretty much the fluids from an infected HIV individual must enter the bloodstream of another individual to spread the infection. For a while, people didn't know exactly where HIV came from, but newer scientific studies have found that HIV may have came from Central Africa, from a type of chimpanzee, and pretty much humans would hunt these chimpanzees, eat the meat, and then that's how the virus was spread from chimpanzees to humans, from humans ingesting the meat of the chimpanzees and spread the virus to humans. Theoretically, that's pretty much what they think. Scientific studies have shown that, and it's a pretty good probability. So the fifth slide is the mechanism by which HIV causes disease. So pretty much any virus you get, like if you get the influenza virus, it turns into the flu, which is where you have, you know, all the symptoms and stuff like that, the sickness basically. Or whenever you are, you know, whenever you're like exposed to a bacteria, and the bacteria, whenever it attacks your body, whatever disease it causes, that's the mechanisms by which it causes the disease, how it works to make you sick. HIV binds to CD4 cell lymphocytes and completes the replication process. So just like we talked about before, HIV will bind to these lymphocytes, and whenever it binds, the HIV triggers a process in the host cell known as apoptosis and increases cell permeability of the cell membrane. Apoptosis is a process of programmed cell death, and all cells can do this, you know, like, you know, in your body, whenever cells have, you know, completed their fulfillment and they've done everything they need to do, and they're not useful anymore, they will actually trigger apoptosis on their own in order to not mutate or cause disease. So this is actually sort of a really cool self-defense mechanism that your body has, but HIV triggers this on healthy CD4 cells whenever it happens. Every time HIV replicates itself, this causes more and more cell death. And over time, you know, that's why it's an extremely slow progressing disease, which is, you know, also why they call it a lentivirus, lenti meaning slow. It takes a very long time to produce, you know, actual sickness from it. This process can take 10 to 15 years, and it eventually leads to AIDS, which is acquired immune deficiency syndrome. And this is essentially whenever your body, this is essentially characterized by an immune deficiency, and it's where your body has little to no immune system because of its attack on these CD4 cells, and your body sort of gets depleted of that, and, you know, hence no immune system, or AIDS, acquired immune deficiency syndrome. The next slide contains signs and symptoms of HIV. Now, HIV can be a little complicated because acute HIV infection, or the first stage, can cause symptoms just like anything else. So like whenever you get the flu and you have like the fever and you feel bad and you feel run down and you have muscle aches and you have chills and a headache and all that good stuff. Not good, but you get my point. it can cause the same symptoms. An acute HIV infection causes pretty much the same things. That's why the only way to know if you have HIV is to get tested. But, you know, it can vary from person to person or stage of HIV progression. So stage 1 or acute HIV infection is characterized by flu-like symptoms such as fever, chills, headache, night sweats, and rash. Some people may not even have symptoms at all, which is, like I said, why it's important to get tested to know if you have it or not. The virus is still multiplying at a slow pace during stage 2, or they call it chronic HIV infection. It's clinically latent, meaning that it shows little to no symptoms. The virus is still multiplying, like I said, but at a very slow pace. And this can take 10 to 15 years before fully progressing to AIDS. And this is stage 3. So stage 3 is characterized by progression to AIDS or acquired immune deficiency syndrome. Symptoms of AIDS include rapid weight loss, extreme fatigue, swelling of lymph nodes, diarrhea, opportunistic infections from badly damaged immune system, and Kaposi sarcoma, which is like a rare cancer that affects the lining of your blood vessels. And this is most commonly shown as little lesions on the face that people get. Kaposi sarcoma is something that we wouldn't normally get because our immune system, believe it or not, actually protects us from a lot of cancers. Whenever you have no immune system, a lot of rare cancers and rare things tend to pop up. The diagnosis of HIV. HIV can be diagnosed by a medical physician using testing resources. These tests may include like antibody or antigen tests that test the blood for antigens of the virus, antibody tests that test for the presence of HIV antibodies in the blood and saliva, and nucleic acid tests that test the blood directly for the virus itself. Any of these tests can be used to test for HIV, but a nucleic acid test is most straightforward directly after exposure because it tests the blood for the virus itself instead of testing for antibodies or antigens. Sometimes it takes the body time to kind of develop antibodies towards a pathogen, so a nucleic acid is the best way to go. But all testing techniques should show some sort of result at least a month after infection, and sometimes they'll test you twice to make sure that a result is correct, given that you've been exposed or you've put yourself into a risky situation involving a sexual partner or something of those sorts. Treatments for HIV. There is currently no cure for HIV. The only treatment available for HIV is a group of medications called antiretroviral therapy, or ART. This treatment option prevents the replication of HIV in the body to achieve an undetectable viral load. Back whenever we were going back to the life cycle of HIV and we were talking about each step, what the antiretroviral therapy does is basically interferes with these stages of the HIV life cycle and stops it from fully replicating and stops the progression of HIV. It's actually a very cool life-saving medication that has caused a lot of people to live a long and healthy life with HIV, because before that there was effectively no hope of living a long life if you got it, because there was no way to stop it. You know, it was unstoppable. Now they have these new treatments that can, you know, that are life-saving, saving lives. It prevents the replication of HIV in the body to achieve an undetectable viral load. It slows its progression, and this allows the individual to live a long and healthy life with HIV infection. People with an undetectable viral load, and an undetectable viral load means that they don't have a lot of virus in their body at all. People with an undetectable viral load due to the antiretroviral therapy pose effectively no risk of transmitting the infection to sexual partners, but the medication must be taken on a strict regimen that's taken daily to allow the medicine to work properly. You can't take it one day and skip the next. That's not how it works. It has to be taken same time every day on the regimen that your physician prescribes to you, and only you, because everyone will be different. That is really the only treatment for HIV, but it saved a lot of lives, and HIV is a big deal, but living a long and healthy life with it, it's not a death sentence like it used to be. Now, ways for you to prevent HIV or HIV prevention: practicing sexual abstinence, which means not having sex. That's hardly beneficial to anyone. entering a monogamous relationship to limit your number of sexual partners, never sharing needles, and using condoms properly every time you have casual sex are all effective tools. Taking advantage of preventative medications such as PrEP and PEP. PrEP meaning pre-exposure prophylaxis, and PEP meaning post-exposure prophylaxis can further decrease your risk of acquiring HIV. This basically stops, if you're exposed to HIV, it stops the virus from attaching to your cells. That way you don't get infected with it. Always getting tested will always, keep you safe, because it always, tells you, it prevents that spread farther. So if you were to get HIV and you follow a testing schedule, if you're not in a relationship and you have a lot of sexual partners, knowing your status can save your life or someone else's, because, if you know that you have it, you're not going to go and give it to someone, Always getting tested, having open conversations about safe sex, and know your partner status as well as yours. Those are all very important. These are all ways to make people more educated about the importance of HIV prevention and cultivate a safer world for all of us. That was the end of my presentation. It ran a little long, but I hope that it was very informative and good. And thank you very much for watching. Bye.